Dear guests, dear friends, welcome. Today we do have people in the room, but we also have people online. And please always remind us that we have people online. In our enthusiasm, we may just focus on the room, but we have people online. And Kristin, who is the organizer of all this, will have an eye on the chat if people have questions, and she will make sure that questions are addressed during the Q&A sessions. Okay, in 2021, KNCV was awarded the Postcode Lottery Dream Fund. It was called No More Pandemics, and it focuses on the use of genome sequencing to accelerate diagnosis and improve surveillance of TB and other infectious diseases and AMR. This symposium gives a flavor of the experience from the Netherlands, using different genome sequencing technologies for surveillance and diagnosis of infectious diseases and the work we support abroad. I hereby give the floor to the KNCV Executive Director, Mustapha Gidado, for his opening address.
Thank you so much, Agnes. Let me join Agnes and other colleagues to say a very big welcome to this extremely important symposium. I think we are really going to hear and learn and understand the value of the entire sequencing system in our laboratory, because I think it is the way forward. Looking at overall the issues related to not only restricted to tuberculosis, but also the entire antimicrobial resistance is becoming a major global crisis. And as we all know as experts, antimicrobial resistance has minimal voice than the other diseases. So there is a lot of discussion, HIV, TB, malaria, and so on and so forth. But if you are looking for a significant voice for antimicrobial resistance, then I think it is a vow or paucity of voice in that area. And I think this discussion is just the beginning of creating a significant voice around it and its importance. And I really would like to thank Kristin and her team for getting us this faculty of colleagues to share their experiences with us. And also, of course, our own staff, Myra, Beatrice, for pulling this together. A very big thank you. And not to say much, I think I'll just hand over the chairing of this meeting back to Agnes so that we can enjoy the session and ask as many questions as possible. We are using this as an entry point into our General Assembly meeting. So along the line, we would also like to invite you to listen to the KNCV performance in 2023 as part of the presentation that is going to be made to the General Assembly after the symposium. So enjoy the day. Agnes, welcome back.
So it's my honor to invite Dr. Richard Anthony, head of the National Tuberculosis Reference Laboratory, the RIVM, to present on the experience with whole genome sequencing for tuberculosis at RIVM and its use for surveillance and prediction of drug susceptibility. And I hope that he will always also enlarge a little bit on the important work you do for Europe. And one more thing for the speakers. The speakers have 10 minutes, have 15 minutes for their presentation and the Q&A session. So I will wave my green flag when we are at the 10-minute point. Yeah.
Thank you very much for the invitation. Thank you everybody who's physically present, and welcome to everybody who's online. It's a long title and I have lots of slides, so I think I'm going to say next slide a lot, and I'm going to try and get through. As I say, as has been already said by Agnes, I am telling the experience of the Tuberculosis Reference Laboratory in the RIVM. And you all know that TB is not the primary public health problem in the Netherlands, but it's still there, and it still deserves attention, and it still deserves monitoring. And I think we can learn a lot from TB control in the Netherlands. I came to the Netherlands because of how strong TB control is, and the fact that you have an integrated system and you have public health integrated, and yeah, that was a big reason. And so I think this is something which you can learn from, may not be appropriate for everywhere, but definitely something to take notice of. And can I advance the slide myself? No. Can you advance the slide, please? Yeah, so we've had quite an extensive experience with whole genome sequencing. In 2016, we already started. And then in 2019, we started using whole genome sequencing officially as our epidemiological typing method. So to rule out clustering between isolates, or say there's a possible transmission, there's a possible epilink, and give feedback to the local health system and allow us to monitor transmission within the Netherlands or imported cases or activated cases from transmission a long time ago. And this is the initial use of whole genome sequencing. And then from 2020, we switched from using phenotypic DST as our primary screen for resistance. We implemented whole genome sequencing as our primary screen for susceptibility. Next slide, please. One thing to mention, we are the reference laboratory, so we get all the positive cultures from the Netherlands sent to our laboratory. So this means we have a good database, so we can do the typing, we can put all the typing data in one place, and we have lots of sequence. We have a low prevalence of drug resistance, which means that we can very efficiently assess the specificity of our pipelines for calling sensitivity. Next slide, please. Yeah, so it's between. We've put quite a lot of effort, and I say this, it seems very boring. Actually, it's the thing that I'm most interested in, so that probably sells more about me than tuberculosis. But anyway, the DNA extraction and what we put into the sequencing is absolutely critical for getting good results and getting fast results and getting reliable results. Because if we have to keep repeating it to get good results, then that also slows us down. So we've put quite a lot of effort in our laboratory, and now Hermans has also been working on that, who will talk later, to getting good input DNA for our samples. And I think, okay, we are working with cultures. We are now extracting DNA from the cultures we receive. We're not subculturing. And I know everybody in the KNCV wants to sequence directly from sputum, which is, of course, the dream. It's the dream funds. But DNA extraction will be an important step in that. And we can all get me too with shiny things. We need the shiny things, but we also need to put garbage in, garbage out. Next slide, please. Whole genome sequencing is extremely good if you have a fully susceptible isolate and you do a whole genome sequencing. You look at the knowledge base of mutations. There's a huge knowledge base of resistance-associated mutations. The TB community can be very proud of that. It's extremely good at calling an isolate sensitive. Next slide, please. These numbers are high, yeah? Percentages are high. I'm trying to go quickly because I'm normally too slow. Next slide. Yeah, and the WHO, as you know, has published a catalogue, the first catalogue for a bacterial pathogen, I believe, which allows us to have a reference document to predict susceptibility from whole genome sequence and also from the targeted sequences that you get from some of the assays we'll hear about. Next slide. This is, I don't know whether this publication will be, this presentation will be shared, but this is a publication explaining exactly what we do in our laboratory and how we apply whole genome sequencing in our process and when we do phenotypic DST. And we do dramatically less phenotypic DST than we used to do. And that's a time saving, a cost saving, so a cost saving, especially because we do whole genome sequencing, or as I already told you, for the epidemiology. So we're leveraging that information to make a more rapid, confident susceptibility prediction. Next slide. I already almost ran out of time, I think. Go. Next slide, please. Oh, yeah. I'm sorry, I'm not lucky. So I should look behind me. Okay, so if we don't find any mutations, our report, which is sent to the laboratory who sends us the isolate, is based on whole genome sequencing alone. Our definitive report is this is an isolate. It's clustered or it's not clustered with another isolate we've seen in our whole genome sequencing database. And if it's predicted to be susceptible to all the first-line drugs, it's susceptible to all the first-line drugs. We will not do any phenotypic DST. This is our definitive report. And I think that what we used to do is say, well, our prediction is this, but we're going to do phenotypic DST, and we'll come back to you on that. And I think that's a bit of a double message. So I think now we have the knowledge base, we have the data, we can really say this, and I think this is very important because we don't want to keep duplicating tests, we want to replace tests, and we want to be more efficient. Next slide, please. That's not to say that there is no role for phenotypic DST, but I think phenotypic DST is more going to be for the difficult cases where you may want to, where you're running out of drugs and you may want to individualize therapy, or to look at emerging new mutations where we don't have a correlation and build the knowledge base for the catalog. And there is quite extensive discussion, active discussion now of what is going to be the structure of this, and that we don't lose the phenotypic DST completely, but we leverage it to provide us with the information that we want. And people are talking about composite reference standards or all sorts of things, but there's a lot of literature out there on that. Next slide, please. Just a quick note to say there is a difference between phenotypic DST, whole genome sequencing, and clinical impact. So when you look at phenotypic DST, you're looking, does my compound kill my bacteria in an artificial growth medium, which I've got? When you're looking at whole genome sequencing, you're saying, do I know something about these mutations? Do I know how this will affect the treatment? And we want to run that back to clinical impact. The catalogs have been produced by comparing the whole genome sequencing to the phenotypic DST. And so this is quite interesting because we're missing out the clinical impact on this circle. So maybe we are propagating some of the limitations from phenotypic DST into the whole genome sequencing. And I think that will be the next development. And this is where we now come to the discussion of maybe sequencing is better at predicting borderline resistance mutations to rifampicin and these types of things, if people are aware of that. So it might not have an effect in the culture, but it might have an effect in the patient. And if the sequence changes, we should be able to see that. Next slide. Next slide. This is some talking about typing. So there's a history of typing. This is Kristin Kremer's beautiful work. This was the most beautiful typing method till 2009. And it was actually very good. It was a lot of work, even more work than whole genome sequencing, I think. From 2016, I've already said we start from whole genome sequencing, and the resolution of whole genome sequencing is really high. Next slide. Keep pressing buttons. Yeah, cool. So if we increase the resolution of our typing method, we should get less false clustering. So more of the isolates we link together should have real epi-links. And press the button again, please. And that's exactly what we see. So with the previous typing method with the VNTR, we had 30% of the isolates we said were linked. There could be an epidemiological link found. If we switch to whole genome sequencing, press again, that goes up to 56%. So for 56% of the isolates, we say we cannot rule out an epi-link. When you actually look, interview those patients, talk to those patients, indeed, in more than half of the cases, it was possible to find a link between those people. Next slide. Next slide. Next slide. Most of the TB in the Netherlands is not due to local transmission. So most of the isolates we see are unique. And that is an ambition. That's one of the ambitions, is to eliminate local transmission of tuberculosis. And to do that, of course, we need to monitor. Yep, next slide. Next slide. This is too technical. No. Yeah, so we are interested in tracking strains of interest, and it's well known there are clones of MDR which are particularly problematic in certain countries, certain regions, particularly in Eastern Europe, and monitoring and tracking these is of great interest, and is not as easy with whole genome sequencing as you might think, because we need to carefully define these clusters. And so there has been a series of EU initiatives on defining these clusters, and we've also recently published a small list of SNPs which can be used to rapidly screen for the presence of these clusters in your country. Next slide, please. You will have noticed from my long talk so far that we're quite reliant on whole genome sequencing at the moment in our laboratory. So quality control is absolutely critical. And this is something we've also put quite a lot of effort in. And also from the perspective of defining what are our limits of detection, what are our noise levels, so we can increase the resolution of our typing, but also more confidently call emerging resistance, or if patients are failing, how quickly can we detect they're failing? Can we detect even that the bacteria are trying to escape the drugs on a failing regimen? That may be possible if we look deeply into the sequence. Next slide. And that was it. And there's a yellow flag as well.
Thank you very much.
Thank you.
Thank you very much.
Did my best.
Yeah. My question, are there questions online? That's for you.
No questions
No
online.
questions online.
Sorry, no.
Are there any questions from the audience? Actually, while others are gathering their thoughts, because you shared a lot of information. One thing you mentioned is actually that the whole genome sequencing is much better at showing transmission.
Yeah. So as you increase the resolution, you're more likely. So yeah. So as the resolution of your typing increases, you rule out more. You see less false transmission. Yeah, that's absolutely true.
Yeah. Yeah, so actually this is interesting also for a program as a means of monitoring the impact of your program, because as you said, and I want to repeat that here, the impact of a program is about interrupting transmission, eventually, and people being cured and so reducing suffering, etc., etc. But that all has to do with reducing transmission.
Yeah, so you would like to see the local transmission drop to zero in the Netherlands, and the only TB we see, well, we'd like to see no TB at all, but first, the one step towards that is to prevent all transmission in the Netherlands, and then every strain we see will be unique or part of a large international cluster.
Yeah. And Richard, I know that you are not only the reference laboratory for the Netherlands, for TB in the Netherlands, but RIVM and your group, to be precise, you are the the European reference laboratory for molecular for genome sequencing. This network is led by Milano, I think.
ERLTB net is called, and this is a network of reference laboratories in the EU. Indeed, it is led by Milan, and we are indeed in the steering committee, and we are responsible for the whole genome sequencing EQA panel.
So without asking an indecent question about performance of others, but I wanted to ask, how far are other countries in implementing this?
Very, very variable. Yeah, I mean and it goes quite quickly, but there are still some countries which have big problems, and the turnaround time is sometimes an issue. But there are different streams of countries, I have to say, and it's also problematic for some countries from the perspective of transmission, because it depends how centralized you are. It depends on the size of your country as well, to be honest. We cannot compare.
Okay, so the centralization is an issue. We will keep that in mind.
It has plus and min. Yeah, it's yeah.
One question for
Yeah, thanks, Richard, for a very interesting presentation. You mentioned briefly that you cannot, or as of yet, it's difficult. It's a dream to use sputum, directly sputum, for whole genome sequencing. So, what is needed to move towards this dream come true?
Well, you can get whole genome sequence from sputum, but not currently from everybody's sputum. So if you have somebody with a very high bacterial load, there are methods, quite complicated DNA extraction methods, and you can get whole genome sequencing from culture. There are some assays in the pipeline coming which claim to be able to go down the amount of bacteria needed, so concentrate those bacteria, get them into your... You've got to get those DNA from those bacteria into your sequencing machine. And that can be difficult, but that is indeed coming. Or the alternative are the targeted assays, and go for the... Because from the perspective of drug resistance, a relatively small proportion of the genome is responsible for almost all of the clinically relevant resistance. That is true.
And that is a good bridge to the speaker after the next speaker. So thank you very much. A great talk, Richard. Thank you very much. So let me now call to the stand Jakko van Ingen. He is a consultant, a clinical microbiologist, and associate professor of medical microbiology at Radboud UMC. And I think that is a very nice combination to bring maybe the genome sequencing into the realm of what it means for patients.
Exactly.
Thank you very much.
I thank you very much, Agnes, and thanks a lot for inviting me to come over. I do accept it as my mission to show that these techniques, and I'm going to say some very bold things, these techniques should be used for direct patient care. That's what we do. These techniques should never be restricted to the TB column. We have suffered in the past from restricting techniques to particular diseases and disease control programs, which in the sense of what I think is really a democratizing, if you can call it that, technique, like next generation sequencing, is entirely wrong. And I'll show you what we do in our hospital to show you why it's wrong to restrict the use of these techniques to reference labs, that's very wrong, and to TB programs, even worse. Which is always nice to say at a TB meeting. So I'm happy that you still made the error of inviting me over. I will try and use the clicker device, although I already heard it's a bit finicky. So I'm aiming at something in general.
Sorry, it has a short range, so you have to do it here. So,
Woo!
Does it work? Yes.
You made it work. So that's good. Wonderful. So if you don't mind, I am going to go back one, because I'm from Nijmegen. And now, as you can see, we are one of the 2 designated TB treatment facilities in the Netherlands, providing TB care for both inpatients and outpatients. And our inpatient program is specifically for those patients who either have very difficult forms of TB, think drug-resistant TB, think TB meningitis, or simpler TB in what we often jokingly call difficult people. Now, these people are not being difficult, but they do have conditions, for example, an allergy to rifampicin, right? That completely changes how you look at treatment of individual patients, even if they have drug-susceptible TB. So we do difficult forms of TB and complex patients who can have either simple or complex TB. So we provide multidisciplinary care. So I'm one of the members of the treatment team, which has pulmonologists, infectious disease doctors, hospital pharmacists, a radiologist, and weirdo microbiologists like myself. And next to our clinical duty, we also are the regional TB reference lab. So we do susceptibility testing by molecular means for several hospitals in our region, as well as the complete package of diagnostics for ourselves. And we are the national NTM reference lab. And I had to say NTM because, and it's one of the reasons why you should think outside TB when you think about applying these techniques. 10 years ago, we crossed into a new realm, and that's the realm of NTM. So if you look at epidemiological studies in the Netherlands, thanks to many people here in the room who have fought hard to decrease the TB burden in the Netherlands, decrease transmission, I think we've been, thanks to your efforts, very successful at it. But where TB disappears, NTM disease will appear. Non-tuberculous mycobacterial disease is what you could call a new epidemic. Mycobacteria related to TB, extremely drug resistant, extremely difficult to treat, cure rates 50% if you do really well, lower in most, and that's tough. And the way you treat these infections is driven by exactly which bacterium it is. So you need really good identification. Hey, welcome to next generation sequencing, which is the ultimate tool to identify these bacteria to the species and subspecies level, which allows you to give good treatment advice. Currently moving into the realm of can you also use NGS for susceptibility predictions? We're not there yet. It's not TB. And again, the TB community, I think, has done a great job at studying how you can predict drug resistance. And that's one of the things that we do. So if we look at our classic algorithm, so this is, patient samples come in to my lab from our treatment facility, our outpatient program, or any of the hospitals that we support. We provide the basic smear microscopy, direct PCRs, targeting the IS6110 for those who love their technical details. And then we set up cultures using both liquid and solid media. And then the clock starts ticking. Based on the PCRs and the smears, we can diagnose TB rapidly within 24 to 48 hours. Wow. But can we predict what's going to be the best treatment for this patient? No. We only know, whoa, it's TB, and it's smear positive or negative. Or we don't know that yet, and we have to wait, and then the clock starts ticking, and we have to wait for culture. And after 1 week, generally nothing. After 2 weeks, many a patient will have a culture positive. And then we sequence. And mind you, we do next generation sequencing, but the whole process takes almost a week because of what Richard Anthony already explained. There's a lot of complex things you have to do before you put stuff onto the sequencer, and then you still have to sequence. 3 weeks after samples come in, we often have a whole genome sequence available, from which we say, okay, it's TB, or it's one of the NTM species, and then exactly which one. If it's TB, we know, okay, which genotype is it? Is it Beijing genotype? Others, no clinical impact, but we tell it anyway. And we get the first real good look at drug resistance mutations. Essentially to all drugs that are covered in the WHO catalog that Richard already explained. So that's technically advanced, but as a clinician, this is irrelevant. It's completely irrelevant because the pace is just irrelevant. This time frame is nice if you're going to do epidemiological typing, perhaps, but not if you're called to advise on treatment, because the correct treatment should be initiated in a matter of days and not 4 weeks. And that's where we're now developing next generation sequencing at our institute. So we do have the gene expert machine. Oh yeah, we use it on the weekends, you know, when we don't run regular molecular tests. So we can predict rifampicin susceptibility within a day in the weekend. On weekdays, we do a line probe assay. 1 day slower, but predicts susceptibility to 2 drugs. Isoniazid and rifampicin, the cornerstones of drug-susceptible TB treatment, right? Rather fast. But in our center, many of the patients get sent to us because there were issues. They have mutations leading to drug resistance. And then we are asked to provide the best possible second-line treatment. And none of these simple assays, you know, expert or the line probe assay, provides you with very good means to then predict what's going to be a good second-line drug treatment for this patient. And that's why we have now started using next-generation sequencing for exactly that purpose. And as Richard Anthony already explained, it's not easy. You need a lot of bacteria to get enough DNA out of them to predict susceptibilities, but we do it, and we use a commercially available platform for it. And I'll get to why that's important for us. Maybe not for you, but for us. In clinical micro, sadly, that's important. What this means is now, if we have a smear positive sample, we do a direct TB PCR and we say, yes, it's TB. Then we go for the next generation sequencing. If it's smear negative, we'll sometimes try, but results are not yet that good. However, the smear positive patients, of course, are the ones who often really need treatment because they have more advanced disease, and at least they are also the ones that you want out of your societies to prevent transmission. But with these techniques, we can now predict what will be a probably successful second-line treatment for our patients with smear-positive TB. And of course, we need to improve these techniques so we can use them for all patients. But all of this is TB, 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 TB. But you know what we do in the same sequencing run? Hospital infection prevention and control. If we see that there's a rise in the number of ESBL-producing Klebsiella isolates in one particular department, we sequence all of them to look for, is this a potential outbreak? We sequence all the NTM. If we have gram-negatives with extreme resistance issues, or typical AMR issues, you know, if we have carbapentamases, we sequence them because we want to know what are these, what types are these. So the sequencer is not for our TB hobby, right? And it has to be used in multiple programs because otherwise this diagnostic is simply unaffordable. So what's the problem? Well, I think there are 2 problems, but there are also a lot of good points, right? So what we need in clinical care of TB patients, but also for other diseases, is speed. Of course, accuracy is quite nice, but accuracy is a luxury and speed is essential. And we need versatility. We need a technique that can do multiple things. We are a clinical micro department. We're not the clinical TB microbiology department, right? So you need a platform that will allow you to run multiple tests at the same time for multiple different issues, because otherwise we are going to charge 1,000 euros for each and every TB patient if we do next generation sequencing. So you have to make sure this technique is widely used. Make sure you advocate it outside the TB program because you need it outside as much as you need it inside. And I'm happy Fabian is here who knows way better than me that you need it outside the TB program. What's the challenge? There are two challenges. Of course, as I said, the financial sustainability is a problem, but there's another threat in Europe, and that is the CE-IVD. As of, I think, mid-next year, all techniques that we use need to be CE-IVD marked in clinical microbiology. And that sounds like dull regulatory stuff, sure. But mind you, most people who employ next-generation sequencing techniques run their own self-made pipelines. And of course, they're brilliant, they're fantastic, they're good, but they will never pass regulatory checks. So we need to look for commercial solutions. That's why we are actively looking for these commercial platforms to do NGS, because that's one of the only ways in which a couple of years down the lane, we're going to be able to convince the European Union that these are good diagnostics that we're providing. Thank you.
Thank you very much, Jakko. And you're doing exactly what you were supposed to do, stirring up a little bit of dust here. So any questions for Jakko? No questions online? Yeah, Natasha.
Thank you so much, Jakko. As always, with a lot of passion, your presentation. May I ask you, can you explain as a microbiologist this phenomenon when the Mycobacteria tuberculosis, the amount decreased in epidemiological situation, non-microbial tuberculosis increased? Why it's happened?
That's a brilliant question. Why do NTM come up in many settings where TB rates go down? The honest answer is we don't know. There are some examples of sort of cross-immunity issues. We've learned, for example, in some Eastern European countries that have stopped BCG vaccination at childhood, that they then almost immediately started seeing lymphadenitis because of NTM in young children. I think there's beautifully documented evidence from Czech Republic, from Slovenia, from Finland. The why is not really clear, but it does seem that at a young age, your BCG might confer some kind of cross-immunity to mycobacterial disease. It doesn't hold to longer age because BCG doesn't protect you against NTM disease at an older age. So the honest answer is we don't really know, but I do think immunity probably is key. Yeah. So if you're not exposed to TB and your immune system is no longer trained in that way to respond to TB, maybe you also become less able at defending against NTM, but no one really knows.
Gidado.
Maybe I can just make a comment, Jakko. Thank you very much for the passion and linking the whole discussion to patient care, but also to alleviate your concern. I think globally the TB program have acknowledged, and there is a serious movement to the concept of integration in general, especially integrated diagnostic platforms. In the TB high-burden countries, it's unlikely for people to keep investing in single tools. And I think there is a serious pressure around the world that most tools, especially at patient level, should go as close as possible in an integrated fashion because of this financial sustainability. People cannot buy a diagnostic tool for TB, for HIV, for this. It's not the way to go. So I think we are totally in agreement with you, and I think this is a common challenge for everyone who is involved in science to see that moving forward, if we are really sincere with antimicrobial resistance in general, then we must be thinking of integrated platforms.
Fully agree.
So big support from the side of KNCV for this. I wanted to bring on one topic which we may want to research is the countries where TB is going down so much as in the Netherlands, that are not so many countries. In those countries you see the NTMs going up. Of course, these are also countries where people with chronic diseases and chronic illness have a longer lifespan or are more prevalent. It's also an elder population. So I don't know at all whether this has been researched, whether you have some insights on that, and otherwise we could do something about it.
Yeah, well, it's hard to do something about that phenomenon, but in general
I mean,
terms,
we could do something about the knowledge
but
gap.
you're absolutely right. So, in general, if TB rates go down, that often signifies that countries are able to invest significantly in the improvement of lives of their citizens, which means improvements in healthcare, improvements in housing. As a result, you see, and sadly, a rise in other diseases, right? You see changes in nutritional status, you see changes in, the prevalence of chronic, mainly inflammatory diseases. And then the emergence of immunosuppressive therapies for those, which indeed then put you at an increased risk of developing opportunistic infections, including NTM disease. Oh, absolutely. I think those two are definitely linked.
Jakko, I would love to continue talking with you, and we'll do that later. Thank you very much. You may give it to Fabian.
Oh, that's better than
so we are moving on to Fabian. Fabian Landman, who's a researcher at the RIVM. And he will discuss with us a different technology which he has been using a lot and exploring in the sequencing of AMR, the use of sequencing for AMR surveillance. Be welcome.
Thank you for your introduction, and also would like to thank for the invitation to be here. And indeed, I will deviate a little bit from TB. I will more be talking about nanopore sequencing and how we use that for CPE or multi-drug resistant surveillance. So why do we sequence at all? We are interested in the spread of carbapenemase producing Enterobacterales and the spread of these strains. And this resistance is usually located on plasmids. These are mobile elements, and so they can also be transmitted from, yes, there's the animation. So the strains can transmit themselves. However, also the plasmids can be transmitted from species to another species. So you can have a very accurate typing scheme where you can tell an E. coli apart from a different E. coli. However, this plasmid could also have been transmitted to a Klebsiella. So you would have to accurately be able to characterize these plasmids in order to tell something if there is a transmission, yes or no, or to analyze these trends. So why use long-read sequencings? Oh, I am instructed to keep this a bit more close to my mouth. So why use long reads? Well, short read only is not sufficient to be able to reconstruct the entire chromosome or plasmids because there are repetitive elements in these genomes. And so these Illumina reads that we use as short-read sequencing technology are usually smaller than those repetitive elements. And also there's also insertion elements, islands, across these chromosomes. So if you have one, the assembler just can't really make something of it. It doesn't know where it belongs on the final assembly, but the chromosome is full with these, so you end up with this spaghetti-looking heap of data. So you can't compare that to something else if you want to distinguish if this resistant has been spreaded from one species to another. So that's why we use long reads. This is another image just explaining the same, how the assemblers just can't really make up what's going on. So you will end up with a lot of loose ends, a lot of positive endings where the algorithm just can't finish it. So in short, how do we generate that data in our institute? We start with a colony on a plate. So yeah, we do have the luxury of having a lot of bacteria as input. We use a Maxwell, so that's really nice for high throughput. We can isolate 48 strains at the same time, very little hands-on time from people in the lab. And the protocol that we use is a rapid protocol by nanopore. So this sequences the native DNA, so there's no amplicon going on. It's a transposon-based complex that is inserting itself into the total genomic DNA. I have to click a lot. And so there's several options that we can load it on. So you can either use a grid ion flow cell or a PromethION flow cell, and the latter just produces so much more data. It's proven very valuable for us because it was difficult for us to reach enough throughput for 48 barcoded strains on a grid ion flow cell. However, the P2 Solo, we think it's amazing, produces a lot more data. And so in short, like I said before, we used the genomic Promega kit to isolate our DNA. We used the barcoding kit with the latest chemistry, the 5 kilohertz. I don't know if it makes sense, but it's the way you want to extract your data at the moment. We also use the latest models. It's also very important because the latest models are the best at translating that electric signal from the nanopore sequencing to the base pairs. For Illumina, we use the NextSec platform. And so at the moment, we are in the process of validating using nanopore only for this surveillance. But I'm mentioning Illumina because of the hybrid approach that we still use for the majority of the species. And so all this data, because that PromethION I was talking about, it produces a tremendous amount of data. It produces about 1.5 terabytes of data in one run. So you have to manage that data. So this is a very complex subject, so I will not really go into detail, but I do really want to stress that this is really an amazing tool that we use for it. It's an open source software. It's called IRODS. It's an integrated rule-oriented data system, and it allows you to manage your data fair. So you can, your findability, your accessibility, your interoperability, and reusability. Basically, it allows you to connect all your sequences to this system, and so when a sequence run is done, the system will automatically see that the run has finished, and you can then set everything else you want it to do. So all our raw data, for example, is written to a tape storage. So that 1.5 terabyte, the majority of which is the raw data, it will automatically be written to a tape storage, so that will reduce the cost. Otherwise, you're paying a very significant amount every year for just storing your raw data. Also, you can start several pipelines of your own interest that you've, well, like Jakko said, yeah, that we write ourselves. You can start them through IRODS and make it do everything you want. You can make it produce reports all on its own. And so, again, this slide is just to illustrate. These are some of the many tools that we run on our data. So the data has been base called using the latest chemistry, latest models, and there's a lot of different tools, open source, available that you can use. Again, these are not commercial platforms. However, Nanopore themselves, they do offer this option, and I saw a flyer that's probably going to touch on that. So, like I mentioned before, we are in the process of validating this for the MRSA surveillance, and we have published a preprint for this where we assessed how well does Nanopore only assemble data compared to the Illumina output. It's a talk on its own. It's a pretty, it's a long talk. We assessed many different species, not just MRSA, also Klebsiella, E. coli, Pseudomonas. In short, it works for a lot of species, except Pseudomonas for us. So yeah, that's something to take into account. If you want to assess how well does Nanopore only perform, you will have to validate this on your own data set. And also, we isolated genomic DNA. So the models still have some trouble with base calling native DNA. So if you do an amplicon-based approach, the models perform really well. So I just want to show a few examples of, what do we actually find out of this data. So this is an example of an isolate with, of a patient who had 2 strains. And so this is the assembled data. So it had a Morganella and an Enterobacter cloacae, and they both contain several carbapenemase genes. And using this method of sequencing, we were able to tell where are these genes actually located. Is there a risk of these plasmids being able to transmit, yes or no? Another example, a patient with just one strain, a Klebsiella, but with two different alleles. They were both located on the chromosome, and the MIC was very high, so probably the rate for transmission not that great, but yeah, it's very resistant. And another example, just to show sometimes there are just multiple copies located on one plasmid. So yeah, probably a plasmid you don't want to see in your patient. Another example, so these are trees constructed from Illumina data where you have a minimum spanning tree and you can see many different Klebsiella being nicely different branches together, and I've highlighted all the strains that carry the gene for BLA-OXA 181. And so we reconstructed one of these plasmids, and we compared it to all these other strains if we found a similar plasmid. And in red, you can now see where we found the identical plasmid. So these strains have the same plasmid, however, genetically, they are very different. And these are some of the examples that we want to see. That's why we use nanopore data in combination with Illumina still. Another example, this is from the preprint that we've worked on. This is also a minimum spanning tree from a previous outbreak of Staph aureus. And in green and red, you can see the short read and long read counterparts. And as you can appreciate from this figure, they cluster together in this figure. That's exactly what we want to see. We want to see the long read only assembly to be identical, or at least to be clustered in the same as the short read would be. So I have a lot of take-home messages I see, but so it is possible to perform this outbreak analysis using nanopore only, even though I didn't went into too much detail on it. You can follow the QR code, which is on the next slide if you want to read more about it in the preprint. Newer base calling models, they are being released all the time, so this is a very difficult subject for the clinical settings. I'm well aware of this. For us as well, we also have to keep validating the newer models. Oh, it actually went to the next slides. Oh, yeah. Right, so as I also said during the slides, it was very difficult for us to reach a 40x coverage step using just the grid-ion flow cells. That's for total genomic DNA. So that's why we use the PromethION flow cells. They have an added benefit of not only producing even more data, but they're also even cheaper. So we're very happy with that. So we can sequence even more, and that's what we love. So those are my take-home messages. And by that, I would like to thank everyone for your attention. And if you would like to read more about the preprint, you can follow the QR code.
Thank you very much, Fabian. I think you served one part of Jakko's suggestion to use genome sequencing for more than just TB. So you're focusing on a multitude of infectious diseases. Are there any questions? From the Kristin? Not from your side. Oh, we can go back to that later. Yeah, I do have a question for you. You have been doing this. Are there other groups in Europe or in the world you know of that are working on this topic?
Absolutely. So we've been working in a European collaboration, was a full force project, where we collaborated with different institutions in Europe that also use nanopore sequencing to assess if we could use this in genomic surveillance of, well, we had the same species of interest, so also the carbapenemase producing Enterobacterales or Pseudomonas aeruginosa or So yeah, there are several groups in Europe and probably the rest of the world too that also use this technique. And broadly speaking, they're on the same board, so they're seeing the same results. They're seeing the models are keep improving. They are really close to being the same as Illumina. The models can produce a different output per species, so that's why it's really important to still assess how well does it perform for every species. And you need to do this for a diverse, like a genetic diverse set of species to validate the model. We believe that is still due to, these models are made by like training a machine learning model, and bacterial DNA is full of DNA methylation motifs. So we think not all those motifs are still present in the data. That's one of our main last few hurdles that we have to overcome.
Thank you very much. Does this, would you say that there is, what would be the place you see in the future for nanopore sequencing as compared to Illumina sequencing in the surveillance of AMR?
Personally, I believe
infectious diseases.
Yeah, well, personally, I believe it will completely replace Illumina. I don't know how fast that will be because the models will have to be thoroughly confirmed. And also right now, we can still compare our output to what Illumina says it should be. So there has to be a way how we can be sure that the nanopore on its own, the data is to be trusted. So we're not at that stage yet, but we're seeing such major improvements, especially over the last year, that I definitely believe it will completely replace it, because not only it's faster, it's cheaper, it also gives you a lot more data, because if you use it on native DNA, you can also run methylation models, so you can actually determine where are different DNA modifications located. So it's a very broad technique, and yeah, I think that is definitely the future.
Thank you very much for your great work and all your insights. So now I'm calling to the stage Kristin Kremer. She's our team lead diagnostics at KNCV Tuberculosis Foundation. We are very proud of the work she's doing, and she will explain that herself.
Thanks, Agnes. Well, the work I'm doing, I'm just trying to put everything in the right directions, but because there are a lot of people working on this project. We have this project to investigate the utility of nanopore sequencing with MinION devices for the diagnosis and surveillance of infectious diseases, not just for TB, but also others. I'm going to try to see how this works. Last night I thought, I will make my slides more, how do you say that? More interactive, and this morning I was regretting that. But the speakers before me have made it quite easy, because you have understood now that genome sequencing is a kind of all-in-one test. You can do for TB and other mycobacteria, you can determine what species of bacterium it is, whether it's drug resistance or not, you can predict that. And also you can do DNA typing and investigate transmission of bacteria. And within our project, we're trying to use this technique for tuberculosis, but also for other infectious diseases by using the nanopore technology. And we have this nanopore device, and Andrii brought it with him. So later, if people in the room would like to see it, they can go to Andrii to have a kind of in-person check on this. But because this technology can be portable and it's rather low cost, we really hope that we can bring this close to the patients, so at decentralized settings. And this is really the aim of our project, to demonstrate that we can use nanopore sequencing with MinION devices to diagnose and TB, but also other infectious diseases such as COVID, and also their drug resistance predicted mutations. We do this in a multi-center demonstration study, including validation study and prospective cohort studies. And this sort of summarizes our approach. In the first part of the project, the past few years actually, we're talking already about, we were really working on generating the conditions in the countries, establishing all the necessary documentation to start the work. Also to develop standard operating procedures for the laboratory and other procedures. And we have in the last year introduced the sequencing technology in reference laboratories. But with this project, we really would like to go further than that. We want to go also to decentralized laboratories. As Jakko already mentioned also, it's really important that this technology can be used close to the patient so it can influence and improve their treatments. Now I'm going too fast. And then also in the project we hope to generate the evidence and the feasibility and the cost to generate the evidence on the feasibility and cost effectiveness of this kind of technologies. So we use different sequencing approaches. You heard already at RIVM they're using, and also I think at the Radboud University, they use whole genome sequencing. And that's actually also what we're sort of doing with SARS-CoV-2. But then to detect that in clinical material, we use polymerase chain reaction, PCR, to amplify actually the whole genome so that it's also detectable in clinical material. For TB, we are starting to use a targeted sequencing approach, which means that we're really targeting certain parts of the genome, mostly the ones that are associated with drug resistance. So if there are mutations in those parts of the genome, they predict whether the bacterium is resistant or not to specific anti-tuberculosis treatments. So for the TB test, we really amplify specific parts of the genome which we subsequently sequence. And then also further on in the project, we hope to use maybe other approaches such as metagenomic approaches. And we have not exactly identified what those will be, but you have also heard that especially in the nanopore technology, things are evolving fast. And so that's also what we are positively anticipating, that these technologies are being developed and we want to use them. Also, I will tell you later also, we are working also in a project to develop a similar a kind of technology which will look at also antimicrobial drug resistance in more general. So we will start with those targeted sequencing assays. And then later on we will go to more complex technologies. So this is, I will briefly go through some main achievements of the project so far, which was an earlier beginning at one of our collaborative centers is the laboratory of Twente Achterhoek Lab MICTA in Hengelo. And also we work together with the RIVM in this project. But the Lab MICTA started already in the first year with SARS-CoV-2 genome sequencing in their laboratory. We had a kickoff meeting and we had a training of trainers first on the midnight kit of SARS-CoV that is used to do sequencing of SARS-CoV-2, the agent of COVID. Then we also had an in-person meeting, which was really nice because we had, of course, this period with COVID in front of us. So it was really good to meet in person, as it is also today to be all with you here in this room. We started with an online training of the people who will work in the countries, and maybe that's an important part I left out, but we plan to implement this project in Kyrgyzstan, Tanzania and Vietnam. So from each of these countries we had people represented in this training. First on the data analysis, because actually we have learned from experiences in the Netherlands from the RIVM when they rolled out the whole SARS-CoV-2 sequencing in the Netherlands, that actually the sequencing is one thing, but also the analysis can be very challenging. So actually that's why we started with that. And in December 2022, we had an in-person training in which people from each of the countries visited the Netherlands to do the laboratory training. And we did that at one of our collaborators at LabMICTA, because actually KNCV, as you know, does not have a laboratory. Then September 2023, it was really like a big accomplishment, because we started the sequencing work in Kyrgyzstan, first with the training, and since then they have been using the sequencing with the MinION devices. We see maybe that Kyrgyzstan is kind of the easy first step, because actually that laboratory is the reference laboratory of tuberculosis in Bishkek. They already had the experiences of using Illumina sequencing in their laboratory. So their laboratory technicians are very proficient already with using this kind of technique, which is not the case, for instance, in Tanzania. But in December last year, we also started sequencing there with the implementation training. And we have noticed over the past months that, yeah, it's maybe not so easy there, but they're really getting there. And of course, they need to get proficient in using this technology. In both these two countries, we are also already really working on the plan for the decentralization. So we've identified the laboratories, and we're really trying to see how to best organize this. And in Vietnam, we are sort of a bit behind with implementing for our project, because there the paperwork and getting all the approvals is a little bit more complex, and has become more complex in the last year, also for other projects. This is just to summarize some of the experiences/challenges that we encountered. I mentioned already some slow administrative procedures. Lengthy importation procedures, because before you start, you really have to make sure that all the reagents and the equipment are at the right places. For now, we are using an early access version of the tuberculosis assay of Oxford Nanopore Technologies, which actually only includes the primers, which is like one of the materials you use in the PCR, in the process to amplify the DNA. And we need to get other reagents that are needed in that process from other companies. And this is also has been complex. But later on, as Jakko also said, when we're starting to use the commercialized tests, which will hopefully come out later this year, then there will be everything in one package, and that will simplify things. We had lack of and non-responsiveness of local distributions. Some of the things that we ordered never arrived, or arrived very late. We had some, in the beginning, the first experiments in Kyrgyzstan, we had some loss of pore activity, so actually the sequencing capacity. We don't exactly know why, but we're still, it has improved. And also there were some practical difficulties, like if there's not enough internet speed, you cannot even update the software to use the machines. But I think on a very positive note, we really showed that it's possible to implement such technologies in such low middle income countries. And that actually, I told you in Kyrgyzstan, they had some experience with Illumina, the Illumina sequencing, and also with the GenoScreen assay to do TB testing. And they perceived actually the Oxford Nanopore Technologies method as much easier. And also if you have better internet, of course, that will solve your downloading processes. In the meantime, WHO has put out a rapid communication. So they are really now also recommending both the D-plex assay and the Oxford Nanopore Technologies assay. They showed it was accurate, cost effectiveness, and also acceptable under these implementable conditions. But also, they also still point out that it's not an easy technology. It's now also in the WHO guidelines, which came out earlier this year in March. Agnes was flagging at me, so I actually need to be quick. But this is also to show you that we have really defined specific objectives in our project, and each objective should really generate the evidence that we really have the aim for in this project. So validation studies are ongoing in 3 of the 4 countries. In Kyrgyzstan, we have really already progressed a lot with looking into the patient-important outcomes of using the Oxford Nanopore Technologies TB assay. We aim to include 743 patients in the study, and we now have, I think, about 150. Also demonstrate the use of SARS-CoV-2 and to develop this all in one test for other infectious diseases and AMR. Well, we are developing this also partly, especially AMR method, in a PAS4AMR project, which I'll come back to later. And like I said, we aim to generate the evidence on the feasibility, acceptability of nanopore sequencing using these devices in the routine settings. And also to determine the implementation cost and budget impact and cost effectiveness. And I forgot to say that for the 4th study, we have already finished, actually officially as of today, inclusion of the patients, because we did that at the same time while including the TB patients. And we have 1,000 patients included already for that. And we have stored the samples, so as soon as we know which technology we'll use, we can just start sequencing. These are 2 other projects we're involved in. A Pan-African network for genomic surveillance of poverty-related diseases and emerging pathogens in Africa, and also a project dedicated to really develop this method for AMR sequencing. And I think this is my final slide. So I told you we have encountered some challenges, which were mostly like on the organizational part so far. But yeah, they were overcome, and this caused a slight delay. But yeah, now we're really in full swing. The sequencing is ongoing in the Netherlands, Kyrgyzstan, and Tanzania. And those projects that we have on sequencing, they are really strengthening each other, we feel. And also the fact that WHO came out with this guidance on the use of targeted next generation sequencing for TB, it will really, well, of course, help our project, but especially most importantly, help the countries to embark on this technology, which hopefully will really improve the treatment of the patients. Thank you for your attention.
Thank you very much, Kristin. I think you really you gave a very clear overview of the work we're doing, and I'm so happy to see that we are really moving into patient-relevant use, decentralized use of sequencing. Are there any questions? Watsi? Yeah. No, sorry, I'll get you this one.
Oh, I saw one here.
Yeah. Thank you, Kristin. Yeah, thank you for the interesting presentation, not only you, but every single other presenter. My question is just direct for every single one. I guess for you, for Richard and Fabian. Yeah, nice to hear that there is an ongoing process of implementing this nanopore. We have seen in one slide of Richard that negative predictive value of this whole genome sequence, especially for quinoline, is around 90%. How could we compare, like, the negative predictive value of this nanopore in comparison to Illumina, or to the traditional whole genome sequence? The fact that why I raised this question is it has been implemented in Kyrgyzstan, where there is high incidence of MDR. That's where there is high trend of using this second line treatment. So how can we just compare this?
So actually for the technologies, in principle, theoretically, it should be the same, because we are looking at the genome and we're looking at certain mutations, and from certain mutations we know like they are very much associated, like 100% almost associated with drug resistance, but from some other mutations it's not yet clear, and especially for certain anti-tuberculosis drugs we do not know yet all the mechanisms that are causing this drug resistance, and that's especially for the new and the recently repurposed drugs. So for those drugs, it's less, the drug resistance prediction is less. Is there something you want to add to that? Because
only that the WHO rapid communication does address this problem, and indeed the validation for different drugs was different for the different assays. So that exactly comes critically to your point. So it depends on the targets included in the assays, and as Kristin says, it depends on our a priori knowledge of the drugs. So the newer drugs, there's more going on than we know about, and maybe those targets are not yet in the assays.
Yeah, and it is indeed true that the Illumina-based method that is now commercially available scored in that sense a bit better than the nanopore-based method. But that's because indeed a certain that version of the nanopore assay did not yet include all the targets, but they have already improved that. So it's looking better and better, I hope.
Thank you. We'll have a last question before we move to the next topic.
Thank you, Kristin, for a really nice talk. I was triggered by the word context, because I think we have lots of techniques, and there's the Illumina and nanopore fight, what is the best technique for the last 10 years or so. And I think it's really now time to have the discussion, what is the context for all these techniques? Because we can generate more and more data. There are mushroom studies that look at every ASBL, to look at whole genome sequencing. We're putting millions of euros in the Netherlands to look at all those data. And also, now we have this technique, how should we use this? Is SARS-CoV sequencing, it's an open question, is SARS-CoV sequencing adding the added value to patients? So I think it's an open invitation to see how we should use these techniques, because I'm a little bit afraid that overusing all these techniques will generate a lot of data, and then we don't know how to use these data. So that's one side note from enthusiastic but critical in the field.
Yeah, that's a really nice one. Thanks, Julian. And I think we could maybe talk about this all afternoon if we make a small group. But maybe to specifically answer your question about SARS-CoV-2, yeah, of course, for the patient, it's maybe not that relevant to know which kind of type of SARS-CoV-2 it is. But I think in general, if we do typing, and that sounds same for TB, we know maybe if cases are related, and or how clones are spreading across the world, and it's teaching us a lot about how these pathogens are transmitted, etc. And certainly, for example, for the Netherlands, looking at surveillance and sequencing every isolate that we have in the Netherlands, which is has been proven to be a very valuable asset. Yeah, and it helps also, in this case, the municipal health service. Okay, we know these patients, they have a similar bacterium. They must have a common source. And if they don't know each other, their source will be out there, and we need to find it to stop the transmission.
Thank you very much, Kristin, for your expose on this. The discussion is not finished. That's very clear. But we'll move to the kind of a demonstration of this interesting technique, which Fabian called it that it's the future. So now we our laboratory expert and WizKid, Andrii.
Hello,
is going to
my name is Fabian Landman. I work as tuberculosis consultant at KNCV.
You want to introduce the video,
Good morning, dear colleagues. Thank you for your time and dedication to our event. And in my presentation, I would like to show the short video which my colleagues Note and Pauline recorded recently, and this video is dedicated to the wet lab procedure, which is taking place when we do targeted genome sequencing for TB using Oxford nanopore technology. So without further ado, I would like to start the video. It will take about 5 minutes. Press. Can you launch it for me, please? Thank you.
Hello, my name is Pauline Lempens. I work as tuberculosis consultant at KNCV Tuberculosis Foundation. Today I'm in the laboratory with Nout Hermans, who works with us as a PhD student. Nout, what are we going to do today?
Today we are going to carry out targeted sequencing of tuberculosis using a MinION sequencing device as the one we have here. With sequencing we can determine the sequence of the DNA which contains all the characteristics of a bacterium, as for example if it is resistant to a specific treatment or to which genetic family it belongs.
Let's see how it's done. We start with DNA extracted directly from a sputum sample from a tuberculosis patient or from bacteria cultured from a sputum sample. We can analyze up to 22 samples in one sequencing experiment. The first step is a multiplex PCR. What is the goal of this PCR now?
Yes, with this PCR we can amplify the regions of the genome in which we are interested in, such as genes that are associated with drug resistance. To avoid PCR contamination, we use various laboratories. One laboratory to prepare our PCR reagents and the other one to add the bacterial DNA to our PCR mix. And that's the same room in which the PCR is carried out.
After 1 hour and 15 minutes, the PCR is finished and we are ready to measure the amount of DNA in each sample and to adjust it to a certain range if needed. To avoid contamination, the amplified DNA is handled in a third laboratory room. We are going to sequence samples from various patients in one sequencing experiment. How do we make sure that we can connect the right results to the patient afterwards?
We are going to label each sample. Every little tube contains DNA from a different patient. And now I'm going to add a barcode to each different sample. After that, I add all samples to one tube. All combined samples together are called a DNA library.
In the next step, the DNA library is cleaned using magnetic beads. The DNA binds to the beads and the beads are separated from the solution using a magnet. In this way, the DNA library can be washed several times without losing the DNA. When the DNA library is clean, it is released from the beads.
The DNA library is now ready to be sequenced. The sequencing takes place in a so-called flow cell, like this one. The flow cell will be put in the MinION device and can be used up to 4 times.
After a quality check of the flow cell, Nout is loading the DNA library into the flow cell. Now the actual sequencing starts.
The flow cell contains a membrane with very small holes, so-called nanopores, through which an electrical current flows. During sequencing, the DNA is guided through the nanopores, which disturbs the electrical current. Each of the DNA building blocks disturb the current in a different way, leading to a unique electrical signal. From the sequence of electrical signals, the computer can determine the sequence of the DNA building blocks.
The progress of the sequencing can be monitored in real time. After approximately 2 to 8 hours, enough sequencing data has been acquired and the sequencing is stopped. Nout, can you show us how the sequencing data is analyzed?
Yes, we analyze the sequencing data with the bioinformatics analysis workflow provided by Oxford Nanopore Technologies. For each patient sample, the results report mentions the quality of the sequencing data, the genetic family line to which the tuberculosis bacteria belongs to, and whether drug resistance was found or not. If drug resistance was detected, then the report also mentions to which drugs and which genes mutations were found. We hope this video provide you an impression about what sequencing entails.
Thank you very much for watching. If you have questions, please reach out to us at diagnostics.team@kncvtbc.org.
So, thank you for watching. I think you got a bit of a flavor of how it's happening in the real life. Yeah, and in your materials you have an example of report printout. That's a real report from the sample which was sequenced in Kyrgyzstan. And most likely it will be in more or less the same shape when M-Pore TB assay from Oxford nanopore will be marketed later this year. Yes.
Thank you very much, Andrii, for this demonstration. Looking at the device itself, it's not easy to organize with a group like this, and not very interesting for the people online who are not here. So during the break, which we are about to have, Andrii will demonstrate for those interested. Where will you sit, Andrii? We have the computer here. So you are welcome to come and ask him any questions you would have, and a demonstration of the device. So I see that you have a question.
Yes, Agnes, I just wanted to say that there have been some comments in the chat. And there was some communication which was really directed to Jakko's talk, and I thought maybe Jakko answered these questions in the chat, but I thought maybe it would be nice also for the audience here to get a summary of that. So, Jakko, can you tell us?
Well, yeah, I think there was one very interesting question which relates to the point that Juriaan just made about, how functional is a tool? What do you do if all your TB diagnostics show no TB, and yet you're convinced clinically this is a TB case, right? Can you diagnose TB in the absence of any diagnostic test telling you it is TB? And, well, yeah, my reply was, yes, you can. Of course, you can diagnose TB even if all TB tests are negative, if you have a strong clinical suspicion, and if you've really done all there is to do, right? I mean, it's not uncommon to forget some very basic things that you can do to diagnose TB. I think a wonderful example is, well, Petra de Haas is next to me now, is the stool diagnostics, which are often forgotten, but can, in select cases, help you diagnose TB even when all other testing was negative. So, yeah. And then the process is you start TB treatment, and if the patient responds well clinically, you may have actually diagnosed TB simply from the fact that treatment led to cure, and therefore it must have been TB. And it's good to realize that TB treatment, at least the combination of antibiotics that we call first-line treatment, is not really good at treating anything else than TB. So if someone responds favorably to that, it's quite likely it was TB and not something else.
Thanks a lot, Jakko. There was one other question, which was about whether TB is transmissible, and also especially with AMR. And yeah, I think many of us in the room have already noticed that indeed TB is transmissible, and also multi-drug resistant TB is sometimes just as transmissible as susceptible TB. And there are really examples, especially in the eastern part of Europe, that there are really big clusters of patients actually who are infected with similar or identical multi-drug resistant TB strains. So that was messages from the chat, Agnes. Over to you. Thanks.
Thank you very much, Kristin. So I would like to ask Dr. Gidado whether he would have one last remark maybe before I close. Or you may leave it to the next. Okay. So, in the first place, thank you very much for the speakers to come to here and to share their thoughts and experiences. I think it has been a very interesting session. And thank you also for really making an attempt, and I think you succeeded in that, to cater for this audience, which is a very mixed audience, not just technical. So I think you did a very good job at that. So thank you for that. I hope that this, or my impression here was really, you gave an overview over not only the techniques and the roles of the different techniques in the field of diagnosis and surveillance of infectious diseases in AMR, but you also showed where the Netherlands is positioned in propagating these innovations and really developing. We are at the cutting edge. We are at the maybe you can say we're ahead of the curve, and we're in the role in the Netherlands, but in Europe, but also globally. And I think that is we're very privileged to be able to play that role. And thank you all who contribute to that, both by doing the research in the laboratories, but also in the clinical settings and also in the public health settings. All these experience, but also the data collection from all these levels of work are really essential in order to keep. Thank you all. We now have a break till 11:30, and please, after that break, we will have a presentation by Dr. Gidado, which is actually the first part of the General Assembly of KNCV. KNCV is an association of associations, een vereniging van verenigingen, and every year we have our General Assembly. And the first address on the work of KNCV will be done by Dr. Gidado, and you're all welcome to stay to hear more about what KNCV is doing. Having said that, thank you very much. Enjoy the coffee and enjoy your demonstration by Andrii.
Let me welcome us back to the second phase of the day. And as was earlier mentioned, this is going to be part of our assembly meeting. And it is my own singular honor to invite the chairman of the Board of Trustees to just welcome everyone and open the General Assembly. Then, of course, I will follow with a presentation. Thank you, Marilla.
Thank you, Gidado. Well, good afternoon to all of you. It's very great to see so many of you joining us for our first part of our General Assembly. In this first part, we'll hear about the activities of KNCV and over the past year, and later on we will go on to the General Assembly more formal points, which is not something that we do with everyone, which is simply part of our Articles of Association and the legal requirements. So very warm welcome, and I hope that you will also give us some feedback on what Gidado is now going to present for KNCV, because we really would like to hear from you. So please allow me to invite Gidado as Executive Director to give his presentation.
Thank you very much, Marilla, and I think we can just proceed with this presentation while thanking colleagues in the earlier morning for the fantastic symposium. In this less than half of an hour, part of this is just to give a little of the activities of 2023, both what is happening globally, but what is the role of KNCV in general. So where are we, what are our achievements, and what is our focus for 2024 and beyond. So that is what I will just spend some few minutes to say. But that is not without first starting by thanking quite a lot of everyone. I sincerely would like to thank all the management team, our frontline healthcare workers, our KNCV staff in the field, our Board of Trustees, our general donors, both institutional and private, because the funding is what makes it available for us to be able to do the mission of KNCV. So really sincere appreciation for everyone who is contributing in our mission. But just to give very brief picture, where is the TB generally in the world at the moment? What we have seen, 3 to 4 pictures globally, is that despite all effort, still there is some level of increase in the incidence of tuberculosis around the world. As of 2022, 2023, the incidence still remains above 10 million. So it is still about 10.6 million persons affected with tuberculosis globally. And of course, we have seen a rise of incidence, but to some extent, we saw a rise of death. But lucky enough, post-COVID-19, the number of deaths is little on the decline. However, when we say little death in TB, it is still talking about 1.3 million persons dying of tuberculosis. So it's still the second leading cause of death after COVID-19, unfortunately. And of course, as based on the global TB report, nearly up to 7.5 million persons have been diagnosed and has been enrolled into TB care in 2022. Is that what we expect? It is a huge recovery from the impact of COVID-19 when we went down as low as 18% of the performance of tuberculosis program globally. That having said that, but there's still really over 3 million persons still missing with tuberculosis around the world. And some general contextual factors is still to show that 8 countries still contribute significant part of this burden globally, but unfortunately they are also the mirror country who are also performing less. So they are also still contributing significantly to the over 3 million people who are not actually being found and being put on treatment in around the world. So they are actually mirror image, meaning that there are quite a lot of drivers that these particular countries are actually sharing in common. And also just to briefly say, of course, when we said we wanted to end TB by 2030, there were some milestones for 2025. And are we in the right direction? Unfortunately, no. Because 2025 is just next year, and if you look at some of the key performance in reducing death, reducing incident, talking about catastrophic cause, the numbers are not speaking good. So the world is still really not doing well. We are making progress, but if you look at these indicators, people affected by catastrophic cause is nearly 50% of people, despite it is said to be free treatment, access to diagnosis, but people still pay quite a lot of resources to get access to this. But reduction in death and reduction in incident, actually, we are just not doing well. The world is just totally not on track. And also on the number of persons being put on treatment, if you look at the drug-susceptible TB, a little more in the right direction, but when we look at drug-resistant tuberculosis, again, the world is just not in the right direction, especially, again, among children. Among children, the world is really, really facing quite a lot of significant challenges that we are only putting 19% of estimated number of children with multi-drug-resistant tuberculosis on care at the moment. That is not good enough. Another major target that we are still actually not on track in general is about TB preventive treatment, especially among household contact. The world is doing very well with putting people on TB preventive treatment among people living with HIV/AIDS, but surely not doing well among actual contacts that are within the household. Few places are claiming to be in the right direction, like if you look at Africa and Europe with the incident reduction, a little in the right direction with better numbers than the other regions around the world, and also including the issues to do with death. If you look at generally issues related to Africa, is not only related to their performance in HIV, because HIV was their major significant driver, and we know that Africa is doing significantly well in access to antiretroviral, and that is what is affecting the incidences at this moment and also affecting the mortality at the moment. Just to summarize around the global picture, I think there are quite a lot of issues still remaining for the world. I said over 30 million persons are still not found and not put on treatment, which is unfortunate. And only 47% of persons are actually getting access to the rapid molecular diagnosis. So the earlier symposium, we are talking about genome sequencing, but even the simpler one, the access to it around the world is still 47%. So the world still has a lot of challenge around the issues to do with laboratory. Of course, the treatment success rate among persons affected with drug-resistant tuberculosis, the performance is really, really bad, which is just about 63% at the moment. And of course, I did allude to the concept that the TB preventive treatment is really low. Unfortunately, this should be our yardstick, considering that we don't have a vaccine. When we don't have a vaccine, TB preventive treatment, as an addition to every other thing we are doing, should have been the major way to go. But unfortunately, the world is not doing well in this direction. Of course, some major challenges that are pushing these issues around the world is not only related to conflict, climate change, and of course, the impact of war. Every interrelated and the complexity in most of the low and middle income countries is extremely challenging. And of course, even somewhere around Europe, there are a lot of issues. In fact, not only, of course, Europe. So if we are not careful, we are creating a lot of access to newer regimen, good treatment options, and so on and so forth for the low and middle income country. But in Europe, because of regulatory issues, that access has also not been there. So I think this is something that we need to pay attention a lot, especially with the rifampicin and also childhood-friendly medication. They are not still available. If you want to treat a very, very small child in Europe, you will do the practice I was doing in 1999. The pharmacy have to crush it and add B-complex or something like that, and I don't think that is the right way to go, at least not for Europe. A low middle-income country can struggle with that, but I think in Europe something needs to happen around this. There are a few opportunities around the world, and we are not going to spend too much time on that. You are all aware that last year there was the UN high-level meeting, which is the second one for tuberculosis, that tried to galvanize political commitment around the world, and it came out with a political declaration, and that political declaration came up with absolute numbers that are very ambitious. So it is an advocacy document around the world that people can push, that if we must end TB, then we must go around the route of 90, 100% access. We need to just ensure there is quite a lot of access. And of course, the last year's UN high-level meeting coincided with two other ones, which is actually universal health coverage and also pandemic prevention and preparedness. And we think these three, they need to start leveraging rather than operating in silos. And again, this is actually alluded to the earlier discussion in the morning, that some level of leveraging and integration concept, people need to start thinking about that. If the universal health system is not working, we might not end TB. And we should make sure that we leverage whatever resources are going to be put into pandemic prevention and preparedness for the interest of TB. Because if it happens, then it is the resources of TB that are going to be diverted to respond to the pandemic, which has actually happened during most of the previous pandemics. And of course, there are quite a lot of other opportunities with funding rounds, with action plan around the Euro for 2023-2030. Of course, the Dutch Global Health Hub could be an option that we can continue to explore. WHO has just established a TB vaccine accelerator so that the journey around vaccine for TB, in the past I used to make a presentation that vaccine is a mirage, but I think the mirage is getting a little clearer, so vaccine is in the horizon, hopefully. But also there were quite a lot of opportunities with price reduction. Right? During the last UN high-level meeting, there were quite a lot of reduction to the TPT if the cost was the problem. So there was quite a lot of cost reduction. And also there were quite a lot of cost reduction to laboratory instruments, especially the gene expert cartridges that are being used around the world. And just coming back from the World Health Assembly, there are other opportunities. Again, WHO is also going to go through another round of an investment launch, because WHO is also having a gap of more than 7 billion to be able to do their own work. So they are going to be looking for resources around the world. And there are quite a lot of updated documents globally, and on that, I think it is, okay, now it is going on its own. But backward and forward is the same. If you can reorganize it, because you see use the same forward, but it still goes through the same. I think there were quite a lot of global policy documents that have actually been launched in 2023, but also most recently in 2024. And I think the next slide, the most recent one was, I think, last week, which is a global guideline on surveillance. There were quite a lot of definition and trying to, again, improve the standardization around tuberculosis. All these are extremely important because they guide our work. Part of our work is providing technical assistance, and therefore we need to have a better understanding of almost all these documents to do our work. And all the other documents are actually available for people to see. Next slide. So now I'll pay a little attention to what we have been doing as KNCV in the last 1 year, in 2023. And this first slide is just a big snapshot of the picture in general. We are privileged that at least in 2023, we are still able to implement above 50 projects around 22 countries. Of course, you may know that in some countries we have our underground offices, while in some places we just provide service based on need. And these projects are range in volume, in size, and on need. Some of them are really project requiring people, and some of them is just providing technical assistance. And of course, as a knowledge institute that we parade ourselves, which we know that is a major role that we are proud to do around the world. Annually, there is a union conference for tuberculosis, and last year's union conference, we were able to contribute over 62 abstract papers and presentations in that union conference. And we are privileged on annual basis, we produce over 40 peer-reviewed papers, and last year we were able to produce at least 54 peer-reviewed articles in different journals around the world. We cannot do this work without income, so our income and revenue generation last year was just above 14 million, and I will come back to it. It's not where we wanted, but at least we have enough resources to run most of the work we do. And of course, most of our staff, over 60%, have direct coverage of funding through the work we do. We are being paid for the kind of work we do. And here you see me mention absenteeism. This was a major crisis for us in the past. And right now, I think work-life balance is getting much, much better within the organization. Of course, this is where we are, our footprint in the different part of the world, whether it is an office or part of our network. Most of you will be aware that KNCV is also in the journey of supporting this concept of localization, ensuring local leadership and ownership of projects. So some countries are already in the lead, making offices, taking the lead, having their own resources. Ethiopia is just on the way. They have finished setting up a board. They have an executive director, and in 2 weeks' time, they will be able to be launching who they are, but they are still using a KNCV brand. And yesterday we got an email from Tanzania. All the ex-KNCV staff in Tanzania pulled themselves together, and they are now trying to launch a KNCV Tanzania. So they are coming together. We just got the email yesterday trying to say, okay, how can they be supported, in which way, and so on and so forth. And we are going to find a way of supporting this journey in general. And of course, this morning, I will not spend a lot of time on this slide. This is the whole issues around the multi-drug resistant TB as part of most of the work we are doing. We are playing significant role in the rollout of the shorter regimen for multi-drug resistant TB in a number of countries. And of course, we are very privileged that there are quite a lot of number of patients already in care, and we have been able to monitor the outcome of some of those treatments. And of course, additionally, we put in technology into the work. Now, again, there is an introduction of digital technology for adherence, especially also among drug-resistant tuberculosis patients in a number of countries. Of course, Kristin and the team have spent significant time in the morning to showcase the amount of work that is being done. Laboratory in TB, it is the cornerstone of the work we are doing, unfortunately, because up till this moment, we don't have a point of care test, so we just need to find our way of continuously strengthening laboratory network as a network, using whichever mechanism to ensure that people just have access to the treatment. And quite a lot of innovation have been actually piloted by KNCV and been pushed to scale at the moment. And of course, a lot of our colleagues are participating in a different platform globally, especially with a lot of EP and program review of different countries. That is, again, where we provide our expertise in ensuring that most of what is being done at country level is data-driven and is actually patient-centered. So we are privileged to participate in many of those places. And also taking TB preventive treatment to the community level. What would be the community role in accelerating uptake to this TB preventive treatment, which is actually still a big bottleneck at the moment. And we are trying to come up with some form of best practice in Ethiopia and Indonesia to showcase to the world. Maybe if we combine healthcare facility and community approaches to scaling up the uptake of TB preventive treatment, that could be a smart way to go. And of course, KNCV is participating in some major global projects, especially the one Smart for TB, which we are part of a consortium of a large number of other organizations, and there are quite a lot of trials. It is really a research project, generally, globally, and we are privileged to superhead some part of this project in some number of countries. And last year, we are very pleased that in some of these projects, our performance really was good enough ahead of even some of our other colleagues in the same consortium. And we are playing a significant role in trying to see what is the environment for the vaccine. If these vaccines arrive, what is the level of preparation? So we need to keep understanding that arrival of vaccine is not the end, it's the uptake. It's the vaccine being either at the arm or in the mouth of an individual that is the case. And that is the bigger difficult science. Of course, not more difficult, but it is a social science that is more complex than a primary science of developing a vaccine, because the uptake and utilization of vaccine can be very challenging in many countries. Of course, we are doing quite a lot of issues around stigma, because we know, again, stigma has still remained a major barrier to care in general. So, of course, we keep providing quite a lot of support in that, including e-courses that people are undertaking. And we are participating quite, again, a lot in trainings together with WHO. There are quite a lot of platforms that different KNCV colleagues participate around the world. And that is the privilege we have, because people acknowledge the level of the work we do centrally, but also what we do in the field. Therefore, if people are developing global guideline and global policy, they want to ensure there is a link, there is a feasibility, there is some form of practicality into the policy that people are doing. And therefore, we are privileged to actually be mostly engaged in most of these platforms around the world. And I think it's a long list that I don't necessarily need to go through it. Of course, I did allude to our contribution to body of science. I did mention our performance at the Union Conference, and I also did mention issues to do around our peer-reviewed journal. We are monitoring this also in line with the KNCV Innovation Pathway to see, are we able to cover as many areas as possible, or is there a blind spot? And if there are blind spots, what is responsible for some of those blind spots? And of course, extremely important is the issues related to financing. Doing this work requires quite a lot of financing. Like any other international organization, we are very, very dependent on institutional donors. Of course, very big thanks to the lottery that gives us also a lot of significant amount of money that is on AMR that can support the mission of our work. We have quite a lot of private donors that make some income, but hugely our work is project dependent. So we do quite a lot of our work on projects. Our budget and estimation for 2023 was about 90 million. Unfortunately, we were not there. We were just able to generate up to about 14 million, which is far below our expectation, and of course also lower than our 2022 performance. If time permits, we can talk about that. Most of you are aware about the global movement of resources for international NGOs. There is quite a lot of this national movement using local entities, using national entities, journey for self-reliance. So most resources are routed through national entities and less through international entities. However, it's not to say that there are no resources for international entities. There are, but it is really restricted to mostly areas about research, innovation, technical assistance, and so on and so forth. So that is the reason why we will see some level of fluctuation in resources. The second one is also the cycle nature of resources. The resources are in cycle. Most of major funding, they come in cycle. And some of these cycles are either 3 or 5 years, 3 or 5 years. So if your particular year is not within that cycle, then you will see a fluctuation in your income. There is a tendency you go back if you align with another cycle of funding, but that is just to understand the context. Having said all this, I think our performance is significantly much better compared to 2022 and 2023, even though here we showed a negative performance of nearly 262,000 within the year. However, within this, already 162,000 of it was already earmarked and funded through some of our funding sources. So our overall negative net is really just around 100,000. And the reason for this is because of good performance on our investment, not only through the project money. Okay, so what has given us quite a lot of benefit was performance of nearly more than 670,000 return on investment on our reserves that we're actually putting into investment. That is to showcase that investment is an extremely important avenue of also generating revenue for an organization to also balance bad years. Because sometimes your bad years can easily be taken care by some of your return on investment. And of course, in some years, it's the other way. In 2020 and 2021, it was the other way. We sank completely in our level of income, but that is the way business operates in general. And of course, like I did say, most of these results are usually appropriated as needed. So this is some of the kind of function that the Board of Trustees will look into, try to agree with it. How are you appropriating this money? And then they will eventually give us their own approval. All these have actually been discussed already. And what we will be doing subsequently this afternoon is just to go into the formal discussion about the approval of these results. Of course, despite all the difficulties, despite KNCV operating in a number of countries, despite the volatility of some countries, our performance from audit perspective has always been good. So that is something that we're very happy and we're very privileged to do that. When you operate multi-country project, there is a chances for running into a lot of financial difficulties. So most of the time, I think in the last number of years, most of our audit reports are actually an unqualified opinion. That is not to say that they don't identify areas that require improvement. So there are quite a lot of areas for improvement that we as management have to respond to, and then we have to show that response to our Board of Trustees, that this is the way we intend to work around some of the risks that were identified by the auditors. But it's good to just say that we think we are doing very well based on the audit and so on and so forth. And of course, one caveat here that is going to be a discussion for the next part of the meeting is that we have used same auditor for a number of years, and we are thinking of a possibility of changing the auditors for some obvious reasons. Of course, resource mobilization is the backbone of what we do, and like I said, is basically more of an institutional donor. Of course, we do quite a lot of private fundraising. The private fundraising in the Netherlands is becoming saturated, is becoming restricted, the competition is getting higher, regulation is getting tighter, so there's quite a lot of learning that is need to be done within the private and individual fundraising in general, while of course for the institutional fundraising is a competition, we just need to keep living up to the game. It is a kind of game that has to do with partnership, with collaboration, and so on and so forth. But it's just good to say that whatever we have written, our success rate is not bad. Our success rate is usually nearly up to 30 to 35% of the proposal that we write, we eventually succeed in them. And of course, moving forward into 2024, there are quite a lot of response that we are doing into what we call relatively big projects, so fingers crossed if we succeed in them. Maybe just last few more slides is just to say again, in the 2023, there were quite a lot of celebration when KNCV became 120 years for a disease that should have been eradicated. So it was actually a reflection. It was not like celebrating that we have still been here. I think it was just a moment for us to reflect that what has gone wrong. A disease that is preventable, treatable, curable, and we have been in this game for more than 120 years. Something is just not working. And again, this is the kind of thing that globally people are all debating around the approaches. Maybe the vertical approach, not emphasizing development in general. Europe and many other people, they were in their way to eliminating TB because of development, not just because of the vertical intervention in TB. So that celebration was re-energizing to us, but it was also a moment for us to reflect on how do we work with people globally, how do we create some sense of urgency, how do we become advocate rather than just only working. So we need to start combining advocacy part of it to create sense of urgency while we're trying to do the technicality part of the work we do in general. And of course, communication is an important thing, because if people don't know what you do, then of course they might not be able to invest in what you do. So quite a lot of work is being done by our communication colleagues, in some of them reaching nearly 1 million persons. We are not yet there. I think TB in general and KNCV secondary, we are timid. We behave like the bacteria, we are nice, we are slow, we are quiet, but that is not the way to play the game. The game is a competition. Nobody cares how good you are. You have to compete. So I think this is where we need to step up our game to operate with the mindset of a business approach. And I think that is something that we're all collectively trying to change our own culture. It is the TB culture in general. And then, of course, added to our own value as KNCV, just do the work. But in this world, it's not just about doing the work. You do the work and you show the work. Because some people don't even do enough work, but they show more than the work. And I think we don't want to be bad people, but at the same time, we really need to advocate the work we do. We need to speak for ourselves. So there's quite a lot of advocacy that is being done globally and also nationally, because we know that here we are also privileged that sometimes through understanding the government system, we will be able to influence certain things into either the Global Health Hub or into the UN level meeting preparation. Another one is coming on antimicrobial resistance. We are privileged that we have some level of understanding into the government system. Does it translate to money? No. It's not yet translating to money. I think that's my discussion with Beatrice. We put a lot of effort, but it doesn't turn into money, but it turns into global good. It is into global good, because if you look at all the amount of work that was done, and then the level of the Dutch investment into global fund in the last replenishment cycle, the money increased by over 13%. The Dutch government contributed nearly over 841 million euros to the global fund. Part of it is due to the advocacy. So it is a global good, even if the money doesn't come directly to KNCV. The rifapentin is an advocacy. We need to find a way to put up our game with the civil society organization that everyone requires the best of the drug. And then we need to find a way of navigating the area of the regulatory environment in general. And of course, we did an assessment with all our major donors. So recently, we set up a small questionnaire and we sent it up front to most of our donors and the partners we work with. And then we engage them. When they have the questionnaire, then we have a meeting with them to see what do they see. How do they assess KNCV and where do they see KNCV areas of improvement requirement? And most of them made this list that, of course, people are still satisfied with the work they pay us to do, so they are very much satisfied with the work and how we deliver what we do. Our technical relevance, people see the technical relevance. If you look at the list of the global platforms that we are all participating in, that showcase that people still want our contribution. And of course, our contribution to the body of science. Some people mention our genuine partnership. Sometimes we go into the partnership with all sense of sincerity that it is about the global good. It is about what we bring to the TB program in general, and not only what comes to us. That is not the mindset of some people into the discussion in general. And people like a lot our concept of innovation to scale. That innovating, demonstrating is not enough. You can only make impact if something have gone to scale. If it is not operated at a scale, you will not be able to make any significant difference to the transmission of TB. And of course, the localization effort, we are not resisting it. We are only saying, okay, what role can we play? How can we support it? And we have showcased that we have gotten 4 successful national entities that we supported, and now they are on their own. They are having big resources. They're making big impact in the TB work they are doing, and I've just mentioned 2 other ones coming. When we had the discussion with USAID, the question was, this is not what they see as a picture from Asia and Eastern Europe and Central Asia. So that could be another area that we can think, what is the way to help create national entities that are strong to do TB work in Asia and actually also the Central Asia region. Of course, there are a few areas that require some significant thinking through. Especially partnership management. Some of them feel that we get the money and then we just go, we just keep doing our work, and then we don't manage the relationship. So that is something that we need to pay attention to. And people question us. You guys, are you researchers? If you are researchers, what type of researchers are you? Are you in clinical research? Are you in operational research? Categorically, some of them said, it is better, Gidado, if you guys can clarify, define who you are in the area of research, and let people know. So that if people are talking, they know that, okay, if we are looking for this, we come to you. But you are not either here or you want to be here. So people raise concern that we might know it very well, but maybe we need to communicate it better to them. That is the questions in general. And the other one that was so interesting is that we need to balance the KNCV ideas and that of the donor. In fact, the word is that don't love your ideas more than the idea of the donor. Because I'm giving you money to do something and you're trying to do what you want to do and not what I want you to do. And I think this is a very big issue, balancing. And that is something that with knowledge people, with knowledge people, if you're not careful, somebody will even critique the call for the proposal. but that is the feedback we got. Try to balance it and don't love your idea more than the idea of the donor. I find this extremely interesting that we need to take forward. And of course, timeline, completion of project within the timeline and communicating reason not to. People cited example with us frequently asking for no-cost extension. And again, there is nothing like that because it might cost something. So I think people don't want us to go that route because it has a reputational and also a financial challenge associated with it. And of course, we need to increase our communication and visibility, which is what I actually earlier also alluded to. We do quite a lot, but I think we need to find a way to sell ourselves. And then again, clearly, this was one comment from one of the major donors to say, you guys are doing quite a lot in the low and middle income country, in the high burden country, but what is going to happen in the Netherlands and in Europe? Considering all the current global crisis, TB is not going to respect these borders, and we see that there are still gaps within Europe. So is there a particular role that you guys can define for yourself actually within Europe and also with the other concept that I mentioned earlier with establishing national entities in some of these countries? And of course, in 2024, based on our approved annual plan, it's as if we have seen some of this feedback because we targeted and we created a team for our 2024 to say that it is a year of efficiency and a year of responsiveness. So it means that we are aware of some of those concerns about delays and projects and so on and so forth. And this year we said we need to make sure that we monitor ourselves very well because we need to really be responsive to the donors and delivering the project. And also we need to make sure that we are actually very, very efficient. Having said that, we are not going to lose sight of the quality of the work we do, but 4 major pillars of our attention, in addition to all the projects we are going to be doing, one is the issue to do with the knowledge, KNCV as a knowledge institute. We need to keep refining. Where are we? What do we want to do? We have been struggling to get educational specialists into the organization because we needed those people to direct this, but recruitment didn't give us a good person up till now, so we are still thinking around this because it requires quite a lot of clarity, a vision about it. There is a goodwill about it, but I think it needs to be done based on where do we see the picture in general. The KNCV network is a nice brand, everybody is moving towards it, but it is not turning into a business model. Up till this moment, it is a goodwill, so there is no return on investment if we do it with people, so we are going to think around that. Resource mobilization is very, very challenging. It's extremely challenging, and we also had a little turnover around our resource mobilization colleagues within the organization, so we're going to think around that. And of course, technology, AI is going to disrupt the way we do the work, whether it is providing technical assistance, whether it is providing training, whether it is visiting. There's quite a lot of changes that are going to happen to our work, and therefore we need to acknowledge that and do that. And of course, we need to review our current strategy. I have presented to the board several times using fancy multiple layers that I was dreaming of, employing people out of The Hague, make them as close as possible to where they are needed because that is where they will do the work better. But I have forgotten about the Dutch labor law and tax law. And now after 2 years, it's not working. It's just totally not working. And if Mario is here, Mario will say, Gidado, it's not working. If we are not careful, the tax people will come back after us with VAT that we have not paid on certain people. So this area requires, again, some thinking through. We are going to put this into our discussion with the board in July. We can provide concrete example where this is, what we have learned over time, and collectively we need to take another decision because we don't need to go that route because the amount of energy spent by the finance and the HR in trying to go through this route is extremely difficult, and I think we just need to rethink it. And then, of course, few areas, of course, is the implementation support for this declaration. And then, of course, global policies and guideline development. This is something that we're still going to be there on most of it. Evidence generation is part of our work. We're mainstreaming it completely. And then, of course, quality assurance and improvement is something that we're going to pay quite a lot of attention in general. And having said that, I would like to say a very big thank you again to everyone who has always contributed to the work of KNCV in general. Thank you very much.
Thank you, Gidado. Thank you, Gidado, for this excellent talk. I hope there are some questions from the audience, and I'm looking around. I at least have one, which is, of course, about the vaccines. What we see in the world is increased travel. We cannot stop the spread of disease. We've seen that with COVID. That's going to happen time and time and time again. So I think, but I'm not a medical doctor, I will say that, but I think that indeed the route to vaccines, and then of course the uptake, etc., is going to be the most important development. Do you see that too? And do you see if the WHO and other organizations, and also maybe commercial companies, because vaccines is something that they can, really make money on, so to speak. Do you see that too? And how far are we ahead of that?
Okay, well, again, I can say that, but I think there are quite a lot of experts. Frank is here, Agnes, and all the team about vaccine. My recent observation is the significant amount of interest and energy in that direction, which was not the case before. So right now, I think there's a global solidarity around it, and I think it is because of the experience with COVID-19 that this is doable. So now there's quite a lot of energy and movement around it. And this movement is not just tokenism. I think there's quite a lot of structural arrangement that people are doing. If you look at the establishment of the WHO Vaccine Accelerator Council, I think that is one mechanism. But I think there is also significant collaboration between Global Fund, WHO, and Gavi in this direction. Of course, there are discussions with the scientists who are working on all the possibilities. The pipeline is not heavy because with vaccine, the pipeline needs to be a little heavier so that you can see the success. The vaccine pipeline is not heavy, but there are quite a lot of potential success. And again, maybe Frank can say a word to it. In 2025, possibly 2027, something could come into the horizon for possible approval, and then we can start struggling with all the manufacturing and the delivery and so on and so forth. Discussion around equity is already coming on board, because if we do the vaccines and we don't address the inequity that we saw during COVID-19, then again, we are too late, because we make the vaccine and then we give the vaccine to people who require it less. So that discussion is all on the ground, but my general comment to you is that there is extreme hope that I have not seen in the last 27 years of my career that I am seeing this year and moving forward. Frank, is that a right assertion?
I think this is an excellent summary of the state of affairs.
Thank you.
Thank you. I'm sure there must be other questions for you, Gidado. I'm calling on the members that are here. I see some of the members here. But, yeah, anyone? No questions? Well, then it was extremely clear, which for me it was, but you know, I've heard it before. So I really would like to thank you then very much. And I would like to thank everybody here. And now we're going to continue with the more closed-off part of our General Assembly. Thank you all so much for coming, and see you.
Yes, so from here, for those that, the members and the BOT, we go to the closed part of the meeting, which is downstairs in the counting room, the telkamer, in that corner generally. And to all the people online, members that are online, please move to the Teams connection, and that has been provided to you. And I want to thank everybody who's been here, and wish you a good journey home.